Welcome to LMP Grand Rounds, everyone. Today's speaker is Dr. Beau Webber, and Beau is an assistant professor in pediatrics in the division of hematology oncology. Beau's an expert, excuse me, in the areas of stem cell biology, hematology, immunology, and genetic engineering. He has extensive experience with genetic modification of various types of cells, and I'm assuming he's going to be talking about some of those today. And he's used targeted nucleases like TALEN, CRISPR-Cas9, as well as cut and paste transposon systems like Sleeping Beauty and Piggyback. Beau received his bachelor's in cellular and molecular biology from the University of Wisconsin and his PhD from the University of Minnesota. I've had the enjoyable opportunity to work with Beau on a project where we're using CRISPR-Cas9 edited tumor infiltrating lymphocytes for the treatment of patients with GI cancers, and it's been really fun. I've learned a lot. This is now a phase 1, phase 2 clinical trial that the cell therapy lab here is manufacturing cells for. And so with that, Beau will speak today on gene-edited T cells for cancer immunotherapy. Thanks, Beau.
Perfect. Thanks, Dave, for the intro. Yeah, so today I'm going to be actually talking about a couple things. One, I'll start out with the kind of talking through the story that led up to the gene-edited TIL therapy that Dave discussed a little bit. And then the second half of the talk, I'll actually talk about sort of, I guess, what you'd call like some of the next generation gene editing approaches we're taking to be able to modify T cells in a multiplex fashion, because I think, to really overcome a lot of the barriers to solid tumor immunotherapy, we're going to need to be able to modify these cells extensively for a number of reasons. So the first half will be kind of the translational aspect, and then the second half will be more on the biology and sort of some next generation applications of gene editing to T cells. So just a quick background on adoptive cellular therapy, or adoptive cell transfer, as some call it. It's I think most are familiar with the chimeric antigen receptor technologies and some of the successes that have occurred in that space. And so, if you think about adoptive cell therapy, what we most think about are a system kind of like this, where T cells are taken from peripheral blood of a patient, and they're either modified to express a chimeric antigen receptor, which is a receptor, essentially a fusion of a fragment of an antibody, an scFv, to an intracellular region that's able to activate the T cell upon antigen recognition in a tumor, or a standard T cell receptor, which actually can be cloned and engineered into a T cell to, again, recognize an antigen presented on MHC in this case. And so these T cells are modified, they're expanded, and then they're infused back in the patient to treat cancer. And this has really seen the most success in leukemia and other hematological malignancies. But solid tumors have been much more challenging to really solve with this approach. So one other method that's been used actually for some time now, and was really pioneered by Steve Rosenberg at the surgery branch of the National Cancer Institute, is the use of what are called tumor infiltrating lymphocytes. And so TIL are actually lymphocytes that are within the tumor. And if a tumor is surgically resected or biopsied in some cases, these TIL can be grown out in specific media with high interleukin-2, for instance, and grown up to large numbers and then reinfused after lymphodepletion. I'll touch on it a little bit later, but another approach is that these TIL can be plated in small fragments, so subsections of tumors, and grown up as sort of microcultures of TIL. And then these can be assayed for specific recognition of specific antigens, then expanded to large numbers and reinfused as well. And so TIL have really shown quite a bit of success, at least comparatively, in the solid and epithelial tumor space, particularly with melanoma And there's been a number of trials here, and this is actually branched out from the surgery branch and is being utilized elsewhere. More recently, there has been some pretty pronounced successes in other epithelial and solid tumors, including cervical cancer, bile duct cancer, colorectal, and recently breast cancer, where complete responses have been achieved in patients with, essentially treatment refractory metastatic advanced cancer. And so what you'll notice is that, one of the things that we notice when we look at this is that this tends to correlate with the amount of mutations, or what you could consider antigens in these cancers. So melanoma has an extremely high mutation rate. But as you move through these other cancers, the amount of mutations is much lower. And so this is where, as I mentioned, the selection for those neoantigen-specific TIL becomes really important, whereas for melanoma, it's largely done without any sort of neoantigen selection. And so you can see that with these cancers, the amount of mutations that are there to begin with are quite low that could be targeted. And so this selection for neoantigen targeting is really important. So in addition to identifying these TIL that can recognize cancer through neoantigens, there's a number of other problems that the TIL therapy faces when it comes to solid tumors. The solid tumor microenvironment is extremely complicated, multifaceted kind of ways in which the tumor can inhibit T cell function. And so, some of these are really familiar checkpoint inhibitors. For instance, PD-L1 is obviously really well known, and that can kind of depend on the environment and whether or not PD-L1 is, that mechanism is really at play in the tumor. But there are lots of other problems as well. Oftentimes the expression of neoantigens is low, or they're poorly presented on MHC. The other thing is that oftentimes these neoantigen reactive TCRs are quite low affinity, and so this can cause issues for T cell activation. These T cells aren't getting like a really robust activation signal. And all of this kind of combines to suppress the proliferation and the persistence of these T cells in the tumor. So, one of the things that we've been really interested in is how we can use genome editing to overcome some of these issues. And so this is kind of a story about one gene in particular, and that this gene called CISH, which stands for cytokine-inducible SH2 containing protein. It's a SOX family, or the suppressor of cytokine signaling family. It's an E3 ubiquitin ligase. And so it had been known for, some time that CISH was able to inhibit cytokine signaling by negatively regulating STAT signaling. And so this was kind of like what everybody thought it did. That was its job. It had been identified for IL-2, 15, and some others. But in 2015, Doug Palmer, who's a collaborator of ours at the NCI, identified a novel role for CISH in the ability to regulate T cell receptor signaling directly. So what he had found is that CISH negatively regulates the TCR signaling by ubiquitinating this PLC gamma, which essentially inhibits downstream signaling and all of the processes of activation following TCR stimulation. And so what they found was that if they took T cells out of a CISH knockout mouse, this is a mouse that doesn't express CISH, they took T cells out of these and transferred them into mice bearing, I think this is a melanoma model. They transferred these T cells in. They saw this profound increase in the ability of these T cells to target and reduce tumor burden compared to wild type here we see in the triangles. And this led to essentially rescue or cure of these mice. Again, substantial improvement compared to wild type. And so I think this was like, really an eye opener, because it was a intracellular checkpoint and it functioned right at the level of the TCR, regulating TCR signaling. There's a bunch more data on that in this paper I'm not going to show here. But the question that came up is, could we actually look at CISH, would this translate over into human T cells? And so in some preliminary work, we looked at, first at whether or not CISH was upregulated upon T cell stimulation like it is in the mice. And what we found was, yes, if you activate these T cells by ligating the TCR, we see an increase in CISH expression. The magnitude actually is, interestingly, it's linked to the differentiation status. So in cells that are central and effector memory, we see a really rapid increase in CISH expression after TCR stimulation. And interestingly, when we looked at T cells from either the peripheral blood or TIL taken from patient tumors, what we found is that CISH was active kind of at a steady state in a fairly large portion of these TIL, suggesting that it may play a role in essentially suppressing T cell function in the tumor microenvironment. And this is just a graphical representation of a number of donors and tumors comparing PBL to TIL. So this seemed like a relatively generalizable phenomenon. And it got even more interesting after that. So we started to do some single cell RNA sequencing of TIL from patient tumors. And what we found is that there's this really interesting, essentially, I guess you'd say mutually exclusive pattern of expression of CISH and markers of T cell activation and exhaustion. And so, this was really interesting, and it kind of set the stage that CISH really is this gatekeeper to T cell activation. You can look at this here in a little bit differently, but again, what you see is that when you look at these different activation markers, the expression is much, much lower in cells that are expressing high amounts of CISH compared to cells that are not expressing high amounts of CISH. We can actually see this at the protein level, whereas if you look at PD1 and 4-1BB, which are markers of activation, they largely overlap in TIL. But with CISH, what we see is that it's largely mutually exclusive. If the cells express CISH, they express very little PD1. And so this raised the question, if we inhibit CISH, can we enhance the function of T cells against tumors? I think it's a really interesting question and a logical question to ask. But the problem is that, how do we do this? How do we inhibit CISH? It's an intracellular molecule. There's no known small molecules to inhibit its function, and you can't target it with an antibody like you can PD1. And so this is where we decided to put on our gene engineering caps and find a way to knock out CISH at the genetic level. And so the way that we approach this is through the use of targeted nucleases. And I won't spend a huge amount of time on this, but just to kind of give you the brief history, the evolution of targeted nucleases started probably now back in 2005 with zinc finger nucleases, which are essentially proteins. They have a arrayed protein array. Each one of these is able to recognize specific nucleic acid motifs. And by fusing these to a FOC1 nuclease domain, you're able to create an enzyme that, when bound on the left and the right side of a specific sequence, the FOC1 would dimerize and create a double-strand DNA break at this target site. And now ZFNs are really hard to make, and they were efficient when you got them, but not everybody could really produce these things. And then actually at the University of Minnesota, the chair of my graduate committee, Dan Voitas's lab, in 2009 developed what are called TALENs, or TAL effector nucleases, which are largely similar, but a little bit nicer because they were more modular. And you could really swap, interchange these modules to recognize a greater degree of nucleic acid sequences to where you're able to essentially correlate it to one amino acid per one base pair of DNA. And this made it much easier to target. But then, of course, I think we probably all know CRISPR came along and really has kind of taken this whole field by storm. And as opposed to using a nucleic or protein sequence to recognize a nucleic acid sequence, it uses a small RNA, guide RNA molecule that recognizes a sequence, a 20 base pair sequence within the spacer region via Watson-Crick base pairing. and it pairs with a Cas9 enzyme. And so when this Cas9 has this guide and the guide recognizes a specific sequence, it opens up the DNA and creates a double-strand break. And this is, now, you can design guide RNAs and have them synthetically made within just a few days to target any sequence of your choosing. And so this really opened up the whole field to move forward quite quickly. And so the main strategies to use a nuclease to disrupt genes are either by knockout by non-homologous end joining, which is the primary repair mechanism that occurs in human cells. So in this case, it's a zinc finger, but it could be a TAL or a CRISPR, makes a double-strand break. And most of the time, this goes through non-homologous end joining, where the DNA gets stuck back together, but then it gets cut again, and it keeps doing that until it makes an error, essentially, and the nuclease no longer binds it. And now that gene, most of the time, the gene or sequence is disrupted by the small insertions or deletions. And alternatively, if you make this break and add a donor DNA that has homology regions, sequences that are homologous on each side, you can undergo a process called homologous recombination. And this can be used to insert genes at specific sites or specifically change base pairs, but typically it's much, much lower efficiency. Whereas in T cells, we're able to knock out genes with 90+% efficiency pretty regularly. HR is much less efficient, typically in the range of single-digit efficiencies. And so the process that I'll be kind of describing that we used in TIL is largely based on NHEJ mutagenesis of the CISH gene. So to do this, the process that we use for T cells is first they are purified or isolated. They're activated by CD3 and CD28, sort of like dynabeads, ligating the TCR with a co-stim signal. And after about 48 hours or so, or a little bit more, in the case of TIL, we use electroporation to deliver Cas9 enzyme as mRNA. So the mRNA, this is mRNA that's modified with specific bases to reduce the amount of toxicity and sort of innate RNA sensing mechanisms. And then we also deliver the guide RNA as essentially as an RNA oligonucleotide that contains chemical modifications on each end to prevent degradation. This is actually a really key criteria to be able to achieve highly efficient editing, is this chemical modification of this guide RNA. And so when we look at CISH, CISH has two main isoforms, and these two isoforms share terminal exons. And so these are the exons that we went after when we targeted our guide RNAs. The first thing we did is kind of take all these potential guide RNAs that were designed bioinformatically, and we used some computational prediction for their target specificity. What we found was that the exon 3 guide RNAs were largely, or the terminal exon were largely, had a higher predicted specificity. And when we took these, designed all of these guides and electroporated them into just primary peripheral blood T cells, we found that the efficiency was highest in exon 3. And we settled on guide RNA 2, which had quite high specificity, also quite high activity. It also cut very close to the beginning of the exon. And this was important because typically with most genes, we want to target early exons, but we just weren't able to do that with CISH because we need to be able to take out both isoforms. So we know that the SOX box and a bunch of other key domains are located in this terminal exon. So targeting upstream can both disrupt the gene from by essentially knocking out the function, but also potentially disrupting this domain, the function of this domain. So now that we had our guide RNA, we did some work in T cells. What we found is that it was really effective at disrupting the CISH protein expression. As you know, as I mentioned, CISH is upregulated upon stimulation. So here we're looking at a Western blot of unstimulated and stimulated T cells. And we see that at the protein level, we're getting about 95+% knockout at the protein level in T cells. So importantly for clinical translation, what we want to make sure is that our CRISPR reagent cuts at CISH and it doesn't cut anywhere else. And so we take two main approaches to do this. The first is just using computational prediction. So this is just sites with that share, I guess, 16 of the 20 base pairs with the CISH target site. And it also kind of looks at their position within the spacer region, and it can spit out sites that are highly likely to be, or not highly likely, but at least predicted to be potential off-target sites. And so we designed PCR amplicons for all of these locations, and then we sequenced extremely deeply, and we were really encouraged and surprised to find that we did not identify a single off-target site, off-target editing at any of these sites. But again, what we're doing is kind of like, we call it the street lamp effect. We're only going to find something where you're looking. And so we also used an unbiased approach called GuideSeq. And so this essentially uses a small oligonucleotide adapter sequence that gets stuck into double-strand breaks randomly, and then it can be used as sort of an anchor for sequencing. And this is a really useful tool to be able to identify unpredicted sites that weren't necessarily predicted by computational or bioinformatics. And so again, we were really surprised to find in 3 independent donors in T cells, which was, this was one of the first times this technique had actually been done in primary human T cells, we did not find any off-target sites. So all of our off-target sites, all of our sites where we saw oligo integration was at the CISH locus. And it wasn't something technical, because we had also designed guides for PD1, and we were able to find off-target site both computationally and also using GuideSeq. So these both picked up the same target site. So the assays were robust. It's just that CISH is a really specific guide RNA in this case, which is good for us. So the next question then is, if we knock out CISH in T cells, does it actually enhance their function? And so we took peripheral blood T cells, we knocked out CISH. This is just looking at protein levels, so anywhere between 70 and 90+% knockout in these T cells. And what we found is that when we activate them through the TCR using anti-CD3 and look at interferon gamma release, we do see an increase, a significant increase in T cell, in the number of T cells that are secreting interferon gamma, which is kind of in line with the mouse, some of the studies in mice. We also find that the polyfunctionality, so I think this is interferon gamma, TNF, and IL-2, we see that the overall, the magnitude of is across multiple cytokines, and there's some increase in polyfunctionality. You can look at this another way. So in this case, what we did is we took peripheral blood T cells and we engineered them with an NY-ESO TCR. So this is a T cell receptor that recognizes the NY-ESO antigen. And so when we took these T cells and we mixed them with a cell line that does not express the NY-ESO antigen, we see no cytokine response, as would be expected, whether or not we just deliver Cas9 with sort of a scrambled guide RNA, or our CISH knockout. However, when we do this and we place these cells in co-culture with NY-ESO expressing cells, we see a dramatic increase in the expression of these cytokines in response to CISH knockout. And if you look at killing of these target cells, we see that this correlates with increased killing as well. So having kind of vetted this out in peripheral blood T cells, the next thing we wanted to do is actually see if we could make it work in TIL, which are quite a bit different. These T cells are relatively beat up and coming from a tumor microenvironment that's been treated with chemo and radiation and other things. So they're a little bit different. And so we had to make some tweaks to our expansion process here. And so this is just a diagram of how we generate knockout TILs. So we still use a CD3, CD28 stimulation step. It's extended a little bit for the TIL compared to peripheral blood T cells. We find that that just kind of helps them gather their footing before we electroporate them. And then typically with peripheral blood cells, we'd electroporate and just expand, but work out of the NCI had developed this protocol that uses co-culture with either autologous or allogeneic irradiated peripheral blood or mononuclear cell feeders. And so after the electroporation, we transfer these electroporated TIL into G-Rex bioreactors with irradiated allogeneic mononuclear cells, and then they're cultured for 14 days. And so as Dave mentioned, this was done over at MCT, and this was just an example of our process validation runs, which went extremely well. So here, just through 3 different runs with TIL, we were able to achieve, right around 90+% genetic knockout by NGS. This correlated with really high knockout of protein. You can see here, and it's also in the Western blot, typically ranging 95% and above. And we saw a small hit in the fold expansion just because of the extra processing we had to do in the electroporation. But overall, through these, we were around 1,000-fold expansion, which is, more than sufficient to be able to generate a clinical product. Importantly, when we took these TIL and we put them in culture with IL-2, what we saw is that the CISH knockout cells expanded much more robustly over time in culture, which would be predicted based on some of the role that CISH plays in cytokine signaling. But importantly, they weren't able to grow without cytokine support. So if we took away IL-2, whether or not they had CISH, they crashed, which is important from a safety perspective. And then again, we actually carried out guide-seq one more time in TIL just to kind of appease the FDA in that regard. And again, found no off-target editing. So from a safety perspective, at least from an engineering standpoint, everything looked really good. So then, as I mentioned, earlier, the neoantigen selection aspect. So our goal was to treat, for the trial, going to GI cancers. And so what we had to do, to go after GI, and then also because of just the way that CISH functions is actually use neoantigen selection. So we wanted to make sure we were putting in TIL that were specifically reactive against the tumor. And so the way this works is that a tumor sample is taken from the patient. Part of the DNA is taken for whole exome sequencing, and the other part of the tumor is grown up into fragments. And after exome sequencing, we identify non-synonymous mutations, which are synthesized as long peptides. These peptides are then pulsed onto autologous antigen-presenting cells, in this case, typically monocyte-derived dendritic cells, but we also use lymphoblastic cell lines derived from B cells as a backup, depending on how many cells we get from the patient. And then we co-culture these TIL fragments with pools of typically 10 peptides each, and look at interferon gamma expression as well as 4-1BB and OX40 upregulation by flow. And using this strategy, we can identify specific fragments that are reactive to specific neoantigens. So in this case, this was a patient at the NCI. These were TIL derived from metastatic, there's a metastatic colon cancer patient. And they ran through this process and they found a fragment with reactivity to this GTF2E1 mutation, both by interferon gamma and also 4-1BB. And so we took these TIL, we knocked out CISH in them. So in this case, it was extremely high knockout rate, as we had seen previously. When we cultured these TIL with feeder cells in the presence of either the mutant peptide or the wild type peptide, what we found again is a very strong upregulation, an increase in the amount of cells producing interferon gamma with CISH knockout compared to the wild type, with very little background activation against wild type peptide, which is really encouraging because we don't want to see that background activation. And just this can be visualized again by flow. We get a much higher rate of these polyfunctional T cells against the mutant peptide when CISH is knocked out. So everything looked very good in the neoantigen TIL. We did this again in another donor, and this one is actually really interesting because this patient had initially been treated at the NCI. It was a sigmoid colon cancer patient. They had identified reactivity to this ALOX5AP neoantigen. And so when this patient, when they drew this fragment out and went to dose the patient after production, they found out in sort of testing that unfortunately has to be done after the fact. They had found that the reactivity to this neoantigen, at least what they could detect in a rep bag, was lost or undetectable. And this happens not frequently, but it happens from time to time where you have a reactive fragment, you rep it massively, and then you find that the reactivity is decreased dramatically, in some cases lost. And so we took a fragment, part of the an aliquot of a fragment. We knocked out CISH, which was a little bit lower efficiency in this particular case, but still pretty good. And what we found was that, again, in the same, similar to what had happened in the therapy, the reactivity to the peptide was lost in the repped cells, but recovered to a certain degree. I mean, pretty modest, but we were to a detectable degree with the CISH knockout. So this was kind of an interesting example where, you know, it could, you know, it sort of suggested that CISH could even sort of resurrect reactivity in a rep product that may have lost or have really low levels of reactivity after the rep process. So we ended up going forward, receiving sort of FDA authorization for the IND to move this forward. And so this is just kind of a breakdown of how it works at the U of M, where again, the tumor is taken out of the patient. It's sent to MCT for fragment outgrowth. So the tumor is cut up and seeded into, you know, 24-well G-Rexes in this case. Part of this tumor is actually sent for exome sequencing. And then we identify single point mutations and do the neoantigen screening of an aliquot of the fragment that's sent to us from MCT. We identify fragments that are reactive against tumor neoantigens, and then we send that information back to MCT, and then MCT pulls out the specific fragments and carries out the CRISPR knockout in the rep, and then they go back to infusion. And on the clinical side, this is led by Dr. Emil Liu, and then in red is, as I said, the MCT with Dave and Darren Sumstead. And then in my lab, in collaboration with Brandon Moriarty's lab, is where we conduct the exome sequencing and the neoantigen screening. So there's a lot of moving parts and a lot of pieces here, but you know, so far it's worked out quite well. So this is just an example. This is one of our, I guess, PV runs for the actual fragment generation at MCT. This is from an ovarian cancer patient, 24 fragments outgrown at MCT, really good growth. We identified 207 non-synonymous mutations and did the screening. We identified 4 fragments that had reactivity here. You're just looking at interferon gamma and then flow cytometry for 41BB. And so we see nice alignment of the interferon gamma and nice reactivity. It's polyclonal reactivity. So MCT pulled these fragments out, expanded them up, 96% knockout, good potency and a little bit higher potency of interferon gamma release in the CISH knockout, which is what we'd expect. And I think this one yielded right around 1 × 10⁻10 total cells. So, you know, this was kind of our first full circuit run of every aspect of this, growing our own fragments from a patient here at the U of M, taking it all the way through the knockout and expansion. And so, so that was a really good sign. So then we initiated the trial in May. This, again, is just an example of a patient, one of the patient screens where we're able to identify multiple fragments with neoantigen reactivity. So everything looking good from GI cancer patients in our hands. And so far, you know, this just recently started. You know, the first patient was dosed in November, and the second patient was dosed in December. And the third patient is set for dosing in the end of January. And so really there isn't any clinical data quite yet. But so far so good. Per the FDA's mandate, we have to do a dose escalation, but the products that we've manufactured so far, the first patient received about 200 million TIL with a 96% knockout of CISH. The second patient received 4 × 10⁹, so about 20 times higher dose, with 92% knockout of CISH. And then the patient that's going to receive the TIL at the end of January, this will be a 3 × 10⁻10 TIL with 95% knockout. So yeah, on the cellular product side, everything looks fantastic. And now we just hope for a good outcome on the clinical side. And so far, I guess the one thing I can say is that the first 2 patients have tolerated the cellular product relatively well with no adverse events. So that's, I think, a really good thing to start. And now we'll just kind of see how things end up on the efficacy side. So that kind of ends the talk about the neoantigen TIL. You know, a lot of some of the things we're still working on are really trying to understand at the basic level how CISH regulates this program of activation. We're kind of digging into the weeds there. We're also working on ways to improve the efficiency of this sort of manufacturing process because it is quite complex. So with that, I think I'll just switch gears and kind of talk about some of the next generation pure editing approaches that we've been developing in the lab. And so, you know, some of this I alluded to, but a number of challenges face adoptive cell therapies, particularly the fact that they're highly personalized and expensive and they're complex to manufacture. Most of them are all autologous therapies. And also, as I mentioned, the delivery of CAR and TCR is required for a lot of these new therapies that are being produced, but using random integrating vectors is not desirable for safety and efficacy reasons, and virus is actually really costly and complicated to produce. And then solid tumors, obviously the antigen specificity and the suppressive microenvironment. So these are kind of, you know, big pillars that are challenges that are being faced during ACT. And so a number of approaches are being taken to overcome these. The development of so-called allogeneic therapies, so engineering immune cells so they aren't rejected by the host and they don't reject the host. Methods to target the integration of a specific CAR or TCR, for instance, knocking it into the actual endogenous T cell receptor locus, and then getting away from viruses. And then I kind of talked about TILs and TCRs, but engineering and checkpoint inhibition in general. And so we're kind of interested in being able to kind of basically take all of these concepts and work them into the next generation of engineered T cell therapies. And so again, you know, we're genome engineers, so that's how we think about this. And so how can we use that to address these challenges? So just to kind of give an example of what you might expect, what you might think of conceptually about a multiplex engineered immune cell product that was able to be given allogeneically to target cancer. And so you could imagine that one aspect would be knocking out a checkpoint inhibitor. In this case, I'm using PD-1 as example, but it could be CISH or something else. Knocking out the endogenous T cell receptor to prevent graft versus host reaction, knocking out HLA expression or something similar to disrupt the ability of the host to recognize and destroy the incoming T cells, and then delivering an exogenous receptor to target the T cells, so delivery of a CAR or a TCR. And so this, you can imagine these are sort of all the engineering steps you would need to do in a cell to make this happen. But the problem is, if you do this with CRISPR, there's a number of risks when you start to break the DNA, and especially when you start to break the DNA at multiple spots. You know, first concern is off-target double-strand break induction. For CISH, you know, it was great because it was a very specific guide RNA, but as you saw, a PD-1 guide RNA had an off-target site. And so the more guides you're putting in, the more targets you're cutting, the greater your chances of having off-target effects. It's also been shown that the cells do respond to this DNA damage, not surprisingly, and it magnifies the more places you cut. And so the other problem is that if you start cutting multiple sites in the genome, these pieces of DNA can stick to one another and create translocations and rearrangements that are not always going to go right back with their the way they were. So these are kind of challenges of using nucleases that we recognized. And so it was really interesting a few years back, I think now 2016, out of David Liu's lab at Harvard and the Broad, they developed this new technology called Cas9 base editing. And so this is based on CRISPR-Cas9, but instead of cutting the DNA, it's fused to a deaminase. And this deaminase, once the genomic site is recognized by the CRISPR-Cas protospacer, this deaminase can target the amination of cytosines to uracil, which is then converted to a thymine. And so you can create these C to T edits at specific sites in the genome without breaking the DNA. And so this was kind of a really a revolution in our thinking about how we can edit the genome, as opposed to just kind of vandalizing it by cutting it and hoping it goes back together in some way that's desirable. You can actually program these nucleotide changes. And shortly after that, they actually evolved another deaminase, in this case an adenosine deaminase, which interestingly does not exist in nature. So there's no deaminases, at least in mammals, that create A to G base conversions. And so they actually evolved this from a bacterial adenosine deaminase. And so the ABEs, as we call them, the adenosine base editors, are quite efficient. They have very, very low indel production and some other benefits as well. So we kind of have these two tools where we can make C to T edits or A to G edits. So we started without making a double-strand break. And so we started to think about how can we use this first to knock out genes? So how can we, leverage this nucleotide conversion to make genes to break their function? And so one way to do that is to actually take amino acid codons and convert them to stop codons. So premature stops, as we call it. And there's like a number of ways to actually do this with cytosine base editors. The other way that we thought might actually be a little bit better is to disrupt splice sites. And this can be done just by targeting splice donors or splice acceptor sites. And we thought that, because there's some evidence in the literature that stop codons can experience read-through under conditions of stress, for example, that disrupting splicing kind of hits the transcriptional level as well as the translational level and could actually be more effective. And so just kind of going back to our example that I gave earlier of this model allogeneic checkpoint engineered T cell, we designed, we tested a bunch of base editor guide RNAs targeting PD-1, the T cell receptor alpha constant locus, and then beta-2 microglobulin. And we went through and tested a number of guide RNAs, and what we found is that largely, in this case, the best guides were splice site disrupting guides, typically disrupting the splice donor of an early exon, but in the case of TRAC, disrupting the splice acceptor before exon 3 was also very effective. We did a lot of optimization up front to overcome some issues with the expression of protein, which we worked out through codon optimization and some other strategies. And we kind of worked our way up the ladder until we were able to get to this iteration where we have, in this case, a cytosine base editor that was able to mediate very efficient base conversion at all of these sites. And this led to really efficient knockout of 3 genes at the same time. So in this case, TRAC, PD-1, and B2M, this is measured by flow cytometry, where we're able to achieve right around 90+% knockout of these genes at the same time in the same cell. And if you look at this using spice plots, just kind of looking at the frequency of cells that have, no edits, single edits, double or triple knockout, you can see that as you drive efficiency really high, you start to get much more uniform cellular products that are triple knockouts, as opposed to sort of like mid-range editing where you get these very heterogeneous cell products, which isn't desirable. So high efficiency editing is really important, especially in multiplex. As expected, the indels, so indels are a mark of double-strand breaks, are very low across the base editors. And actually they're the lowest with our most efficient editor enzyme. And that's actually kind of explainable by the more protein you have and the more efficient the way the base editor works, the more reduction in double-strand breaks, which is a good thing. And so what we were really interested in is, okay, if we take away the double-strand breaks at these multiple sites, do we actually eliminate or decrease the frequency of translocations? And so when we looked, this is just looking at a circus plot showing all the different chromosomes. These are all of the different translocations that can occur if you start cutting the genome in multiple spots. Our 3 sites, and in this case, there was 1 off-target site for PD-1 at low level. And so we designed a bunch of droplet digital PCR assays to measure these, and we found that indeed, when you're using Cas9 nuclease, we detect basically every single one of these possible translocations, balanced translocations in this case, in the cellular product. Some of these up in the 4 to 6+% range, and that, like I said, that's just considering the balanced translocation. But what was really striking was the fact that we were not able to detect any of these translocations when using base editor, which was really, really exciting and really encouraging for us. And this actually translated through to the even the growth of the T cells. So if you just look at their fold expansion, when we start knocking out 3 genes, we see a reduction in the actual growth of these T cells. But with base editor, we don't see it. These cells grow almost as good as if you had not even electroporated them. And I don't have the data here, but we found that as you add more guides, this kind of keeps going down, but it's not the same with base editor, where the cells seem to handle it much more in stride. And so when we edit these cells, they maintain like a typical profile. In this case, we had our multiplex edited cells that we put in a CAR using a lentivirus. They typically have a profile that's the same as a cell that just was pulsed and didn't receive mRNA. They secrete cytokines really effectively. This is just looking at your standard battery of cytokines. They're polyfunctional compared to the multiplex edited cells compared to pulse. And they kill target cells really effectively. So here, this is just looking at a CD19 CAR killing of Raji or Raji cells overexpressing PD-L1. And so you can see that we see enhanced killing with the base editor, especially if the cells are expressing PD-L1. Just kind of looking here, these are control cells where even at 3:1, they aren't able to achieve statistically significant killing, but the base edited cells are able to kill these PD-L1 expressors about the same as a wild type Raji that isn't expressing PD-L1 as would be expected with PD-1 knockout. So kind of the last thing I'll talk about, and this is where I'll leave off the next step. So as I mentioned, now we can knock out the multiple genes, but one of the other things we want to do is be able to take a CAR or a TCR and put it into a specific spot in the genome to overcome some of those issues with randomly integrating viruses. And so the way that we wanted to do this, while still keeping the number of double-strand breaks in the genome to an absolute minimum, is to utilize the base editor as a knockout and then utilize a different nuclease system. So in this case, what would happen is the guide RNA is actually a different structure. So the guide RNA could not be interchanged between the two, which would mean that the base editor can be targeted to knock out genes. Basically, its specific task is knocking out genes. And the orthologous nuclease would actually just target one specific spot to trigger the knock-in event, in this case by homology-directed repair. And so, as I mentioned, HDR or HR is really inefficient compared to NHEJ. And so one of the ways right now is to use recombinant adeno-associated virus, or RAAV. And so we have a system that we use to kind of optimize this, which knocks in a GFP reporter, in this case at the AAVS1 safe harbor locus. And so the only way we see GFP is if this GFP lands here. It has a splice acceptor site. It doesn't have a promoter itself. And when it does that, it splices into the AAVS1 gene, and this drives expression of GFP, which we can detect by flow cytometry. And so with Cas9 nuclease, what we find is we can get about 80% knock-in in primary human T cells. And then we tested a couple other orthologs, in this case, Staph aureus Cas9, and we were able to find that with enough optimization, we were able to get pretty appreciable levels. And then CPF1 or Cas12a protein, in this case, delivered as a protein, we were able to find this ultra version that was actually really efficient at mediating knock-in at AAVS1. And so what we were really interested in is knocking a CAR, a TCR, into the T cell receptor alpha locus. And this had been shown by Michelle Sadelain's group to enhance the function of these T cells because the receptor was actually regulated like the normal endogenous T cell receptor. And so we identified guide RNAs for Cas9, SA-Cas9, and CPF1 that can mediate really efficient knockout and cutting at track. So this was promising. So what we then did was design a strategy where we took Cas12a protein to drive our knock-in. And in this case, it was a knock-in of a mesothelin-specific CAR at the track locus utilizing AAV6. And then the base editor would take out, do its sort of normal thing and knock out genes. In this case, we added CISH into the mix, because we're becoming interested in that as well. So 4-gene knockout with a single gene knock-in, CAR knock-in at track. And what we found is that we can achieve really efficient knock-in of this CAR at the track locus when we do it by itself in the 50 to 60 plus range. When we knock out 3 genes, or when we knock out all 4 genes, it doesn't disrupt the efficiency of knock-in at this site. And at the same time, we achieve really efficient knockout of the genes, so TRAC, B2M, PD1, and CISH. So 4 genes knocked out, 1 gene knocked in with very high efficiency. And what we found so far, this is still some early data, but these cells kill really effectively. This is just co-culture with an A1847 ovarian cancer cell line. And even at 1 to 3, 3 to 1, we're basically max killing. And even at 1 to 3, we see good killing. And we're kind of stretching this out a little bit. We're going to stress these T cells a little bit more, because what we're interested in is you can kind of see, if you squint, a little trend of these 4-gene knockout, which obviously includes the CISH, killing a little bit better. And I think if we stress this a little bit more and force the T cells to have to kill, a number of target cells, we might see this this actually magnify. So with that, I'll just kind of end the summary of our what we call our hyper-multiplex engineering with the base editing system. So I kind of showed that base editing is really efficient. Splice site targeting is most effective. We actually just had a whole nother manuscript that's really interesting on this, looking at disrupting all of the components of the T cell receptor, all of the CD3 chains, with a whole battery of, I can't even remember how many guide RNAs it was, I think in the 100 plus, really characterizing premature stop codon versus splice site targeting. And Mitch Klusner in the lab actually developed a web tool called SpliceR, where you can go and put in your favorite gene, and it'll spit out a bunch of predicted base editor guide RNAs to target splice sites to help do that. And that was just actually accepted now the other day at Nature Communications. So that was pretty cool to hear. And now we're moving to where we can do the combined knockout and knock-in. We have it working really highly efficiently. And so the next steps here are, completing in vivo studies, which we're scaling up for right now. And then really kind of moving into this hyper-multiplexing. So how many genes can we edit with base editor? And the other thing we'd like to do is move beyond simple knockout. So instead of just knocking the gene out and taking away its function, we think that there's probably some really interesting things we can do by manipulating like protein domains, by changing amino acids, or by causing splice or exon skipping. So I think we have some creative ideas there about how to really make these T cells highly functional and immune to sort of suppression in the tumor microenvironment. And then lastly, our knock-in right now, we're using a virus, AAV, but we're doing a lot of work to be able to knock in cargos without virus. And I think, when we get to that point, we'll have a methodology to really make some highly engineered T cells that we can hopefully carry forward and someday test in clinical trials. So a lot of people working on this. Obviously, I collaborate really closely with Brandon Moriarty on all of this work, and Scott McIver on the TIL and some of this non-viral engineering work. And a lot of members in the lab have contributed. I'll go through all their names. Dave and Darren at MCT have been awesome. Our collaborators at the surgery branch, Steve Rosenberg, Nick Restifo, and Doug Palmer on the TIL and the CISH knockout. UMGC, Tim Starr has been really awesome helping with the ovarian TIL, and many others as well. So with that, I'll stop and leave a few minutes for questions.
Thanks, Beau. That was great. Like I said, I learned something whenever I talk with you. I always learn something. You do some amazing work. Let's open it up for questions. We do have a few minutes. Does anybody have any questions for Beau?
Yeah, I'll start, Dave. This is John Crossman. Fascinating talk. I really appreciated that. obviously, whenever you start doing genetic engineering, there's concern about some serious adverse reactions, and Rosenberg has pretty much always had some reactions and adverse effects when he's been using his TIL cells on the patients. So the question I have is, with the 3 patients that you're talking about, and you're currently are treating using the CISH knockout, what do you tell them are the reactions that they might have? And in particular, is there concern about cytokine storm?
Yeah, no, I mean, those are definitely were kind of like the top concerns in our mind going into this. Some of the evidence that there's a couple things why we felt that lessen the concern. One is that the CISH knockout animals, so if you have knockout of PD-1 or CTLA-4, those animals actually have some pretty profound autoimmune pathology. But the CISH knockout mice don't have that. They have a little bit of maybe some like I guess like asthma or some airway inflammation, but it's nowhere near the same as what you see with PD-1 and some of those other checkpoints.
Yeah.
So it seems to be limited to acting when there's specific T cell receptor engagement. So that was kind of one line of evidence. The other thing is that, as opposed to treating somebody with melanoma where there's many, many antigens, and we're using bulk TIL, we are neoantigen selecting. So the antigen repertoire of the T cells that are going in is focused a little bit by neoantigen selection. So those are kind of 2 things that we think help with that. But that being said, I think that it still was a major concern. And so anytime you're going to be putting in large amounts of T cells, you have a problem, you have a potential for cytokine release and sort of, fevers and all of those sort of things that come along with it. And so that's why the FDA really wanted us to start out with a pretty low dose of TIL. So 200 million TIL is quite low. And now we're ratcheting up, and so far it's looked good. The other thing too that sometimes, these patients get a pretty heavy Cy/Flu regimen before they're dosed with the TIL. And then they get high dose interleukin-2 infusions afterward periodically. And so sometimes dissecting what's coming from the high dose IL-2 and what's coming from the TIL can be kind of difficult. And one of the things we do is, we give the IL-2, but if there's any sign of sort of, the patient not tolerating it well, we stop that aspect of it, because it's, there's been complete responses with TIL where they haven't gotten IL-2. So it's kind of something we do, but it's stopped if there's any sort of pathology. But certainly, these patients are watched very closely, because I think, that is one of the things we're trying to vet out is whether or not knocking out CISH does exacerbate a potential cytokine release syndrome. So it's to be determined, but so far so good. But now we're getting up into some real substantial doses of TIL, and we'll have to see what happens.
Do you have any way of tracking the TILs that you're giving into the tumor?
Yeah, so there's 2 ways we're going to do that. One is by parsing out the antigens that they're recognizing, and we can identify the T cell receptors and then produce tetramers. That's kind of the classic way to do it. We take blood from the patient at periods after infusion, and then after 6 weeks, they actually get an apheresis, so we get a large amount of blood. But what's really unique about the CISH editing trial, and that's not been possible before, is now we can actually do deep sequencing of peripheral blood lymphocytes, and we have essentially a genetic barcode in those TIL. And so we're going to be able to track the frequency of those TIL in the blood molecularly, in addition to be able to look at the T cell receptor. So I think we're really excited about doing that. And we actually have some pretty kind of cool ideas about how we can use single cell sequencing to basically capture the TIL that are barcoded by CISH, and then look at T cell receptors and gene expression in those TILs. So really excited to do some of that work.
That's neat.
So very nice, talk, Dr. Webber. I quite enjoyed that. My first question is pretty much piggybacking on John's question, and I may have missed it. So, but how does CISH downregulate the action of T cells? It doesn't look like it's at the level of antigen presentation, so it must be somewhat
Yeah.
And then I have the second question quickly, so that then concerning your trials, what are your endpoints, and what exactly will you be looking for to say if this worked or not?
Yeah, so that CISH is actually functions, it's T cell intrinsic. So it actually, after the T cell recognizes antigen, CISH becomes upregulated inside the T cell receptor. And normally it would polyubiquitinate PLC gamma, which then inhibits sort of downstream T cell activation. So it actually, it functions inside the T cell itself on TCR avidity, or antigen recognition by the T cell receptor.
Sorry, yeah, I know, because we're running out of time. I didn't want to miss this opportunity. So in that case, how do you regulate the function of CISH itself?
Yeah, so I mean, in this case, we essentially permanently disable it with the CISH. So with the CRISPR knockout, so we edit the DNA, and then the protein is no longer made in those TIL. So it isn't regulatable right now, although that would be really cool someday to be able to do that, but it's just not something we can do right now. In terms of the trial, so I'm more on the engineering side. I'm not, Emil Liu is kind of running the trial side, but typically I think, it's just going to be sort of RECIST criteria for solid tumor. Hopefully the tumor shrinks and goes away, I guess, bottom line.
Okay, thank you for joining us today. We are out of time. Thank you, Dr. Webber, Dr. McKenna. Yeah. Everybody be well, and we'll talk to you next week. Thank you.
All right. Thanks everybody. Yep.
Thanks, that was great. Take care.